I hope you have seen my previous videos, the first video on the photosynthesis electron transport chain, and the second video on the possible origins of photosynthesis. I will make a fourth video on the Calvin cycle of photosynthesis that makes the glucose sugar. First, I must focus on a key enzyme of the Calvin cycle, the Rubisco enzyme. There is a controversy about how good or bad Rubisco is at its job of adding an inorganic CO2 to the organic molecule RuBP. This task is the first stage of the Calvin cycle to make glucose. The controversy surrounding the Rubisco enzyme is complex enough that I decided it should get its own video. And this third video in my series on carbohydrates and photosynthesis is dedicated to Rubisco. The light-dependent reactions of photosynthesis take place in the photosynthesis electron transport chain of the thylakoid membrane. The light-independent reaction of photosynthesis is the Calvin cycle. The Calvin cycle takes place in the stroma of the chloroplast. The Calvin cycle is also called the dark reactions of photosynthesis because it requires no light photons. Do not let the dark name of the reaction fool you. The dark reactions, aka Calvin cycle, occurs during daylight and simultaneously with the light-dependent reactions in the thylakoid membrane photosynthetic electron transport chain. The photosynthetic electron transport chain provides the NADPH and ATP molecules that power the Calvin cycle. The Calvin cycle uses the energy from NADPH and ATP to convert carbon dioxide, CO2, and water, H2O, into the simple sugar, glucose C6H12O6. Glucose molecules are stored in long-chain glycogen molecules that provide medium-term energy storage for all cell functions. The glucose molecule can provide the basic building block for all other sugars, as well as fats, proteins, and nucleic acids. So, glucose production in the Calvin cycle is key for the survival of all aerobic life on Earth. Before I can flesh out the Calvin cycle, I feel I must dedicate a full discussion to its key enzyme, Rubisco. My purpose is to show there are two sides to the controversy surrounding the goodness or badness of Rubisco. Rubisco is the enzyme that catalyzes the first stage of the Calvin cycle. Rubisco is a large protein. Hans Walter Helt and Birgit Piachulla write that Rubisco contains 470 amino acids. Emre Sevindik records that Rubisco proteins are between 166 and 477 amino acids long and have atomic masses between 18,700 and 52,800 Dalton units. A Dalton unit is 1/12 the mass of a carbon-12 atom, or about the mass of a hydrogen atom. Whatever its true size, Rubisco is a complex protein made up of hundreds of amino acids and thousands of atoms. My first unanswerable question is, how does such a complex protein as Rubisco come to be created? Rubisco is criticized for being too slow and too inaccurate. It is slow. It catalyzes around 3 reactions per second. It is inaccurate. Rubisco's job is to add a carbon dioxide to RuBP. Sometimes Rubisco makes a mistake and adds an oxygen to RuBP to make phosphoglycolate. Phosphoglycolate is a useless molecule. The phosphoglycolate must be stripped of the falsely placed oxygen by other enzymes to make it a useful organic molecule once again. Speed and accuracy are the two main criticisms of Rubisco. A third criticism of Rubisco is that to make up for its first two apparent shortcomings, plants must produce a boatload of Rubisco. And so this makes Rubisco the most plentiful protein on Earth. And this production of Rubisco wastes a lot of organic matter that might have better uses if not for Rubisco's shortcomings. I feel I must discuss whether these criticisms of Rubisco are justified or misplaced. To discuss the topic of the criticisms of Rubisco, I will rely on testimony of Rubisco critics. And the testimony of those who think Rubisco works well within the expected norms for a catalyst given the difficult task of fixing carbon dioxide into organic carbon. In the end, I hope to convince you that criticisms, though accurate, are misplaced and taken out of context, and that Rubisco works as good as can be expected for an enzyme given such a difficult task. The much maligned enzyme Rubisco is the most important enzyme in the Calvin cycle. The name Rubisco is a shorthand for ribulose 1,5-bisphosphate carboxylase oxygenase. Rubisco is maligned for its poor catalytic efficiency in many important publications, including Robert Tabita and Thomas Hansen's paper, short-titled Evolution of Rubisco. Tabita and Hansen claim Rubisco with a turnover frequency around 5 per second that is among the lowest for any biological catalyst. I agree with Tabita's turnover frequency. I disagree that it is the lowest for biological catalysts in its class. There are catalysts in the Brenda database that are as slow or slower. Tabita continues. The bona fide Rubiscos, forms 1 to 3, form a coherent clade, suggesting that they share a common line of descent. Tabita continues. The base of clades containing all known bona fide Rubisco sequences suggests that this clade may have originated in the archaea domain and subsequently been distributed to bacteria, eukaryotic algae, and higher plants. Tabita continues. The Euryarchaea harbor the deepest branching Rubisco and RLP sequences, which therefore makes them the best candidates for the evolutionary route of the Rubisco and RLP superfamily. I find Tabita's tracing the origins of Rubisco back to archaea fascinating. There are no photosynthetic archaea. Archaea do not use the Calvin cycle. Yet, archaea do use Rubisco for fixing carbon. And serious studies suggest the Rubisco enzyme originated in the archaea domain and then spread into the bacteria and eukaryote domains, possibly through genetic transfer or endosymbiosis. The bashing of Rubisco for its slow catalytic activity continues on the Protein Data Bank website, PDB 101. According to the website PDB 101, quote, in spite of its central role, Rubisco is remarkably inefficient. As enzymes go, it is painfully slow. Typical enzymes can process 1,000 molecules per second, but Rubisco fixes only about 3 carbon dioxide molecules per second. Plant cells compensate for this slow rate by building lots of the enzyme. Chloroplasts are filled with Rubisco, which comprises half of the protein. This makes Rubisco the most plentiful single enzyme on the earth, unquote. PDB 101 also says, Rubisco often mistakes O2 for CO2, and this fault results in phosphoglycolate instead of the intended phosphoglycerate, and the phosphoglycolate causes additional wasted steps. Quote, in Rubisco, an oxygen molecule can bind comfortably in the site designed to bind to carbon dioxide. Rubisco then attaches the oxygen to the sugar chain, forming a faulty oxygenated product, unquote.
Has a turnover number of about 3 molecules per second. This is a very slow enzyme. It's one of the slowest enzymes you'll ever run across. Compare it to some enzymes like catalase, which has a turnover of 40 million per second, and compare that to 3. Okay, so Rubisco is very slow.
The YouTube channel Catalyst University compares the slow 3 reactions per second of Rubisco to the ultrafast enzyme catalase, which can catalyze reactions at a turnover frequency of 40 million per second. In context, the catalase enzyme has the simple task of breaking down hydrogen peroxide H2O2 into water H2O and oxygen O2. As anyone who has used hydrogen peroxide as first aid disinfectant, oxygen bubbles up out of hydrogen peroxide naturally. The catalase only speeds up a naturally occurring process. In contrast, Rubisco has the daunting task of adding a carbon dioxide to the carbon chain RuBP, so we would expect Rubisco to act much more slowly. The final nail in Rubisco's coffin comes from the popular YouTube channel Crash Course. I love the fast-paced style of the Crash Course channel. Unfortunately, the Crash Course video on photosynthesis went too fast with its bashing of Rubisco. Crash Course did not give any context to the difficult job Rubisco has in adding a carbon dioxide to the 5-carbon chain RuBP molecule. Adding a carbon dioxide to RuBP is not simple. Crash Course overstated the slow turnover frequency of Rubisco as very unusual. Crash Course overstated the inaccuracy of Rubisco. Lots of enzymes make mistakes. Crash Course harped on the toxic waste products. Lots of processes result in toxic waste products. How toxic is a matter of how quickly the waste is removed.
The Calvin cycle begins in the stroma, or the empty space inside of the chloroplast, if you remember correctly, and this phase is called carbon fixation because, yeah, we're about to fix a CO2 molecule onto our starting point, ribulose bisphosphate, or RuBP, which is always around in the chloroplast because not only is it the starting point of the Calvin cycle, it's also the end point, which is why it's a cycle. CO2 is fixed to RuBP with the help of an enzyme called ribulose-1,5 bisphosphate carboxylase oxidase, which we generally shorten to Rubisco. I'm in the chair again. Excellent. This time for a biography of Rubisco. Once upon a time, a one-celled organism was like, man, I need more carbon so I can make more little me's so I can take over the whole world. Luckily for that little organism, there was a lot of CO2 in the atmosphere, and so it evolved an enzyme that could suck up that CO2 and convert inorganic carbon into organic carbon. This enzyme was called Rubisco, and it wasn't particularly good at its job, but it was a heck of a lot better than just hoping to run into some chemically formed organic carbon. So the organism just made a ton of it to make up for how bad it was. Not only did the little plant stick with it, it took over the entire planet, rapidly becoming the dominant form of life. Slowly, through other reactions known as the light-dependent reactions, plants increased the amount of oxygen in the atmosphere. Rubisco, having been designed in a world with tiny amounts of oxygen in the atmosphere, started getting confused. As often as half of the time, Rubisco started slicing ribulose bisphosphate with oxygen instead of CO2, creating a toxic byproduct that plants had to deal with in creative and specialized ways. This byproduct, called phosphoglycolate, is believed to tinker with some enzyme functions, including some involved in the Calvin cycle. So plants have to make other enzymes that break it down into an amino acid, glycine, and some compounds that are actually useful to the Calvin cycle. But plants had already sort of gone all in on the Rubisco strategy, and to this day, they have to produce huge amounts of it. Scientists estimate that at any given time there are about 40 billion tons of Rubisco on the planet. And plants just deal with that toxic byproduct. Another example, my friends, of unintelligent design. Back to the cycle. So ribulose bisphosphate gets a CO2 slammed onto it, and then immediately the whole thing gets crazy unstable. The only way to regain stability is for this new 6-carbon chain to break apart, creating 2 molecules of 3-phosphoglycerate. And these are the first stable products of the Calvin cycle. For reasons that will become clear in a moment, we're actually going to do this to 3 molecules of RuBP. Now we enter the second phase, reduction. Here, we need some energy, so some ATP slams a phosphate group onto the 3-phosphoglycerate, and then NADPH pops some electrons on, and voila! We have 2 molecules of glyceraldehyde 3-phosphate, or G3P. This is a high-energy, 3-carbon compound that plants can convert into pretty much any carbohydrate, like glucose for short-term energy storage, cellulose for structure, starch for long-term storage. And because of this, G3P is considered the ultimate product of photosynthesis. However, unfortunately, this is not the end. We need 5 G3Ps to regenerate the 3 RuBPs that we started with. We also need 9 molecules of ATP and 6 molecules of NADPH. So with all of these chemical reactions, all of this chemical energy, we can convert 3 RuBPs into 6 G3Ps, but only one of those G3Ps gets to leave the cycle. The other G3Ps, of course, being needed to regenerate the original 3 ribulose bisphosphates. That regeneration is the last phase of the Calvin cycle. And that is how plants turn sunlight, water, and carbon dioxide into every living thing you've ever talked to, played with, climbed on, loved, hated, or eaten. Not bad, plants.
The Rubisco controversy is not about human freedom or legal rights. It is poor taste for science to attack religion. It is poor taste for religion to attack science. Attacks start arguments that cannot be resolved. Crash Course gave no examples of a better way to fix carbon than Rubisco. I don't criticize Crash Course for echoing the opinions of respected research scientists that Rubisco is slow and inaccurate. I will reference the opinions of respected research scientists that Rubisco is not as slow and inaccurate as other enzymes in its class. I will criticize Crash Course's mockery of intelligent design. To refer to something that God made as unintelligent design is an assault on religious people, many of whom are well-educated scientists, engineers, and medical doctors. I don't worry about educated people being able to filter out the bias. I worry more about students and younger people accepting bias as fact. There are opposing opinions to the slowness and inaccuracy of Rubisco. The poor functioning of Rubisco should not be used as proof there is no God. That religious argument cannot be resolved with Rubisco. The poor functioning of Rubisco is taken out of context. Results of study by chem015e et al. At the Australian National University show that Rubisco works as well as any other catalyst in its class. I will take a moment to quote from the Crash Course comments about Rubisco. At about the 9:30 mark of its photosynthesis video, Crash Course goes into an aside on Rubisco. The aside describes the origin of Rubisco a couple of billion years ago. Crash Course describes a little organism that needs organic carbon to grow. Crash Course says, quote, "Luckily for that little organism, there was a lot of CO2 in the atmosphere, so it evolved an enzyme that could suck up that CO2 and convert inorganic carbon into organic carbon. This enzyme was called Rubisco," unquote. So far, so good. At the 9-minute 55-second mark, Crash Course says, quote, "And it was not particularly good at its job, but it was a heck of a lot better than hoping to run into some chemically formed organic carbon, so the organism just made a ton of it to make up for how bad it was," unquote. Now, it is true that organisms that need it do make a lot of Rubisco. It is more controversial to say that Rubisco is not particularly good at its job or that it is bad. Context is left out. Crash Course does not name another enzyme that can fix carbon faster than Rubisco. There are no other natural catalysts that can do the job of Rubisco. At 10 minutes and 5 seconds into the video, Crash Course continues, quote, "Not only did the little plant stick with it, it took over the entire planet, rapidly becoming the dominant form of life. Slowly, through other reactions, known as the light-dependent reactions, plants increase the amount of oxygen in the atmosphere," unquote. At 10 minutes 18 seconds, Crash Course continues, quote, "Rubisco, having been designed in a world with tiny amounts of oxygen in the atmosphere, started getting confused. As often as half the time, Rubisco would start slicing ribulose bisphosphate with oxygen instead of CO2, creating a toxic byproduct that plants had to deal with in creative and specialized ways. This byproduct, called phosphoglycolate, is believed to tinker with some enzyme functions, including some involved in the Calvin cycle. So, plants have to make other enzymes to break it down into an amino acid, glycine, and some compounds that are actually useful to the Calvin cycle," unquote. And all of this is true. Crash Course continues at 10 minutes 50 seconds. Quote, "But plants had already gone all in on the Rubisco strategy, and to this day they have to produce huge amounts of it. At any one time, scientists estimate there are about 40 billion tons of Rubisco on the planet, and plants just deal with that toxic byproduct. Another example of unintelligent design," unquote. The Crash Course statement referencing unintelligent design is a swipe at the intelligent design theory. A theory that life and the entire universe was created by an intelligent designer, God. The writers of the Crash Course channel do not take into account that many of their viewers might believe in a creator God. Crash Course takes a cheap shot against believers in intelligent design without making any effort to look at reasons why an intelligent designer might deliberately put a limit on the speed or accuracy of a design, or allow it to do bad things along with good things. That Rubisco can and does create toxin when it mistakenly adds oxygen to RuBP instead of CO2 does not disprove the intelligent design theory. Plate tectonics and movements of the Earth's crust do bad things like cause earthquakes and volcanoes that kill people. But crustal movements do good things like mountain building and the upwelling of minerals from deep inside the Earth. There might not be any dry land without crustal movement. the Earth's surface would be eroded flat and covered by the ocean water if not for plate tectonics. There would not be fresh nutrients to support life if not for plate tectonics. Without the good and bad of crustal upheaval, the Earth would be as dead as the Moon or Mars. So bad things can have a good side if examined with more depth and when put into context. There are many reasons, including flexibility and adaptability, that not all biological processes have machine-like precision. All biological processes produce byproducts that, if not removed, will become toxins. When we use our muscles anaerobically, we generate lactic acid that must be removed. We have a liver and kidneys to remove toxins from our blood. Bacteria, and all cells, have ATP synthase-like proton pumps, or electron transport chains that pump out protons to prevent the buildup of protons that are released during normal biological activities. These protons would acidify a cell's internal fluids if the protons were not pumped out. Toxin management is a normal biological activity. The existence of toxins does not prove there is no God. Sometimes it is more economical to make a mess, finish the job, and clean up after the job is done, than to take care not to make a mess in the first place. Sometimes it is more economical to make do with a less precise machine that makes a lot of rejects, but gets the job done, than to spend money on an expensive precision machine that makes no rejects, but never pays for its original cost. It may be cheaper to keep driving your old car than to buy a new car that gets a little better gas mileage. Rubisco has been around since the first photosynthesizing bacteria over 3 billion years ago. If another competitor organism came up with a better carbon-fixing molecule, that competing molecule would have driven Rubisco into extinction long before the first plant incorporated the first chloroplast ancestor into itself between 1.8 billion and 600 million years ago. That did not happen, so we can conclude that Rubisco is adequate for the job it does. Rubisco is as good or better than any carbon-fixing enzyme that came before or since. Natural photosynthesis is not as efficient as man-made solar panels. Solar panels can convert about 20% of the sunlight falling on them into electricity. This electricity could be used to split water to make usable hydrogen. From the heating value of this hydrogen, we could obtain its energy content versus the light energy used to make it to get an energy efficiency. A solar panel can turn about 10% of the light energy falling on it into usable hydrogen, according to David Biello in a Scientific American article, Plants versus Photovoltaics. By contrast, quote, photosynthesis conducted by algae turns roughly 3% of incoming sunlight into organic compounds, unquote. According to photonics.com, quote, in leaves in full sun, up to 80% of the absorbed energy must be dumped to avoid its diversion into toxic chemical reactions that could damage or even kill the plant, unquote. There is a big difference between solar voltaic cells and plants, according to Michigan State University researcher David Kramer. Quote, this is a big difference because if photosynthesis makes a mistake, it makes toxic byproducts that kill the organism. Photosynthesis has to be conservative to avoid killing the organisms it powers, unquote. Despite all its inefficiencies, photosynthesis gets the job done. Photosynthesis has kept plants and photosynthetic bacteria growing and multiplying for over 3.4 billion years. If perfect design was the goal of the designer, human muscles would be as strong as machine hydraulics. Humans could lift objects weighing many tons, and our legs would allow us to run faster than a race car. If perfect design was the goal of the designer, birds could fly faster than the fastest man-made supersonic jets. Since biological systems are only sufficient to allow the creature to live its life, that might be the designer's plan. I will leave it to one of my references, Camille Bathelier et al., to discuss the specific facts about Rubisco and whether it is good or bad or neutral at its job of adding carbon dioxide to RuBP. Camille Bathelier, Guillaume Cherkez, George H. Lorimer, and Graham D. Farquhar published an article called Rubisco is Not Really So Bad in the Wiley Online Library on January 28, 2018. Camille Bathelier did this work at the Research School of Biology of the Australian National University, Canberra, Australia. At this writing, Camille Bathelier is at the Ecology Laboratory at the University of Paris. I have excerpted parts of Camille Bathelier's article for my purposes of showing a counter-argument to claims that Rubisco does a poor job of fixing carbon. Camille Bathelier's report is many pages long and includes many tables, charts, and graphs that I will not include. Camille Bathelier and his co-authors call Rubisco's turnover frequency modest, not slow. Quote, because of its relatively modest turnover rate, a few catalytic events per second, and the competitive inhibition by oxygen, Rubisco is often viewed as an inefficient catalyst for CO2 fixation. Considerable efforts have been devoted to improving its catalytic efficiency, so far without success. By comparison with other chemically related enzymes, we find that Rubisco is not especially slow. Furthermore, considering both the nature and the complexity of the chemical reaction, its kinetic properties are unremarkable. It is more likely the result of a compromise between chemical and metabolic imperatives, unquote. He continues, quote, important details of the catalytic process remain unclear. First, the sequence of proton transfers and exchanges involved in the reaction is still incompletely understood. Second, the origin of the reactivity with O2 is unknown. Third, the reasons for the limited turnover of Rubisco and its promiscuity, i.e., its propensity to catalyze other reactions, are not well identified, so that both sluggishness and confused specificity are now tacitly accepted in the literature, with limited consideration of practical constraints on the mechanism. In addition, despite 50 years of intense research efforts, attempts to improve the catalytic performance of Rubisco have been so far unsuccessful. This simply emphasizes the need to better understand basic questions associated with Rubisco catalysis. The knowledge of precise mechanistic constraints during Rubisco kinetics will aid current research for a better enzyme. Unquote. She continues, quote, in practice, the justification of comparative adjectives such as slow and confused remains limited. In reality, how slow and non-specific is Rubisco? Using comparisons with other enzymes, we will show that Rubisco A is not particularly slow, B has a good affinity for CO2, and C is not exceptional in being promiscuous, unquote. Camille Bathelier references Brenda, a database of over 2,000 catalysts. Quote, database Brenda of kinetic parameters revealed that the median turnover frequency is 10 per second, with 60% of values within 1 through 100 per second. Therefore, Rubisco's K catalyst value looks rather unremarkable. That is, not particularly low, as Barr even et al. Pointed out, unquote. She continues, quote, Rubisco's K catalyst, per K metabolic, falls between 0.5 times 10 to the 5th and 5 times 10 to the 5th per meter second. That is, in the mid to upper range of most frequently observed values, unquote. Camille Bathelier continues, quote, as a result, mutations that enhance catalytic parameters but disfavor stability will be counter-selected, unless destabilization is sufficiently modest to allow compensation by subsequent stabilizing mutations. The stability-activity trade-off in protein evolution induces epistatic effects, leading to irregular evolutionary landscapes and evolutionary dead ends sometimes encountered in directed evolution experiments, unquote. Camille Bathelier continues, quote, enzymes operating close to equilibrium, i.e., with an in vivo Gibbs energy of reaction very close to zero, such as fumar-ACE and TPI. In this case, the thermodynamic driving force will be the main factor controlling the flux in a given direction, so that the apparent rate of the reaction in vivo will be substantially lower than the maximum turnover measured in vitro, unquote. Camille Bathelier continues, Quote, Rubisco belongs to a specific category of enzymes catalyzing virtually irreversible reactions, i.e. with a strongly negative Gibbs energy. It is the entry point of carbon into the Benson-Calvin cycle, so that it is often considered as the rate-limiting step RLS for the carbon assimilation flux, unquote. Camille Bathellier continues, quote, note that the concentration of Rubisco sites in a chloroplast approximately equal to 4 millimolar is about 300 times the concentration of CO2 in equilibrium with air. Carboxylation efficiency of Rubisco cannot be considered as being poor. It rather seems to have a catalytic efficiency that matches metabolic imperatives, and B is similar to that occurring in other reactions of primary metabolism across most living forms, unquote. Camille Bathellier continues, quote, a large number of enzymes can catalyze more than one chemical transformation. This phenomenon, referred to as enzyme promiscuity, is now considered as being the rule rather than the exception. It is believed to have played a central role in the evolution of enzymatic functions across several protein superfamilies, because promiscuous activities can rapidly be enhanced with a limited number of mutations, unquote. Camille Bathellier continues, quote, Rubisco is not known to catalyze any significant reaction on a substrate other than RuBP, and is thus not promiscuous for the sugar phosphate substrate, unquote. She continues, quote, by contrast, the oxygenase function of Rubisco is a clear case of promiscuity, with a catalytic efficiency only 100 times lower than that of carboxylase. This might be viewed as an unusual case of promiscuity, because promiscuous functions are expected to provide a physiological advantage that can be selected for under appropriate conditions, unquote. Camille Bathellier continues, quote, the reason why it has not been subsequently eliminated during evolution then remains unclear, unquote. Camille Bathellier continues, quote, enzymes producing similar types of carbon ionic intermediates during catalysis should also exhibit O2 reactivity. Presumably, Rubisco catalyzed carboxylation is accompanied by a competitive reaction with O2 for stereochemical reasons related to the primary sequence residues and the influence of metal coordination. This conclusion is probably broadly applicable to all carbon ion-forming enzymes, irrespective of whether they involve a metal ion, unquote. Camille Bathellier continues with his conclusion. Quote, taken as a whole, the present survey of enzymes and comparisons with Rubisco's chemistry show that Rubisco is neither slow nor unspecific. It behaves as do many other enzymes with a similar mechanism, unquote. Camille Bathellier continues, quote, of course, there may still be potential for improvement, but the protein has evolved to respond to a multiple chemical compromise, catalyzing the complex multi-step reaction of fixing CO2 onto RuBP in an oxygenic atmosphere at physiologically compatible rates. Such a catalytic efficacy probably explains the overwhelming success of Rubisco as the prevalent carboxylase across all eukaryotic and prokaryotic taxons, unquote. Camille Bathellier asks the question, quote, is a better Rubisco desirable, and would it improve plant photosynthesis, unquote. Camille Bathellier continues, quote, targeting a supposed RLS enzyme overexpression and/or desensitization has been attempted several times for many metabolites in plants, starch, lipids, amino acids, vitamins, but unfortunately, it has never met significant success, unquote. Camille Bathellier continues, quote, Calvin's cycle involves large fluxes, so that it has to be tightly regulated to avoid rapid and huge metabolite accumulation and divergence from steady state, unquote. Camille Bathellier continues, quote, therefore, targeting Rubisco alone might not be as efficient as often anticipated in increasing photosynthetic fluxes and growth in field conditions, unquote. And so, I have presented chem015e and his colleagues' conclusion that Rubisco is not so bad. I hope this presentation successfully counters against the general consensus of the scientific community that Rubisco is slow and inaccurate. When compared to other organic catalysts, Rubisco falls within the normal range for both speed and accuracy. Rubisco's promiscuity in catalyzing undesirable reactions is common to other catalysts. Promiscuity among catalysts is the rule, not the exception, and may provide evolutionary or adaptive advantages in unknown environments. All the hype around Rubisco's sluggishness or confused specificity is out of context. These behaviors are normal or unremarkable compared to other catalysts. If anything, Rubisco is less promiscuous than other catalysts. For what Rubisco does, it is not particularly slow, and its turnover frequency falls within the normal range for most catalysts of its type. The biases against Rubisco in the scientific literature reflects hope in the mind of researchers that, with a little genetic modification, the speed and specificity towards carbon fixing, rather than oxidation mistakes of Rubisco, can be improved to the great benefit of agriculture and all humankind. Researchers at Lancaster University in the United Kingdom have published their efforts to improve Rubisco for agriculture in a phys.org article titled "Fickle Sunshine Slows Down Rubisco Enzyme and Limits Photosynthetic Productivity of Crops." The subject of their study is the cowpea, a crop plant in Africa that is imported for its high protein content. The Lancaster scientists have discovered that photosynthesis slows down when clouds block the full sunshine from the cowpea fields. And it takes photosynthesis about 20 minutes to gear up to full activity once the cloud passes and full sunshine returns. They believe delay in activity can be traced to delays in the Calvin cycle in Rubisco. They estimate that on days of intermittent clouds, the delays in the return of the Calvin cycle to full production can reduce the carbon dioxide uptake of the cowpea crop by up to 20%. The Lancaster researchers found that cowpea Rubisco deactivates after only 5 minutes in the shade. There are thousands of varieties of cowpea. They are studying different varieties of cowpea plant to discover if they can pinpoint which varieties have a slower Rubisco deactivation rate. If they can find a cowpea variety with a Rubisco type that remains active for longer periods in the shade, say 7 to 10 minutes, the cowpeas will not be as affected by a short duration of shade during a passing cloud. If the Rubisco deactivation rate is slower, they believe the delay in the restoration to full carbon dioxide uptake can be reduced. If a cowpea variety with a slower Rubisco deactivation rate is found, that cowpea variety can be crossbred into local varieties of cowpea crops grown across Africa, and so improve the protein yield across the African continent. If successful, the Lancaster research team hopes the technique of studying Rubisco deactivation rates under intermittent shade conditions could improve the productivity of other plants and agricultural crops throughout the world. The Lancaster research team is hopeful. As of this writing, they have not found Rubisco types with a slower deactivation rate. Other agricultural research scientists also study ways to improve Rubisco. Two research collaborators at University of California are Patrick Shi, assistant professor of plant biology at UC Davis, and Professor Jill Banfield of UC Berkeley. Their research is documented in an article on the UC Davis website written by Greg Watry, August 31, 2020. Professor Shi is quoted, "Although there is significant interest in engineering a better Rubisco, there has been little success over decades of research. Thus, understanding how the enzyme has evolved over billions of years may provide key insight into future engineering efforts, which could ultimately improve photosynthetic productivity in crops." Unquote. In a nutshell, it is chem015e's professional opinion that Rubisco is not slow for the reaction it must catalyze, and Rubisco's promiscuity and inaccuracy is normal for most catalysts. Professor Shi acknowledges that despite decades of engineering effort to improve Rubisco, there has been little success. Plants must produce boatloads of Rubisco to produce the amount of sugar they need for themselves and the rest of us. Compared to other enzymes in its class, natural Rubisco is not so bad after all. It is a shame that some scholars must criticize nature for minor inefficiencies like speed or inaccuracy, and strive to exploit these flaws as a political tool. These scholars cannot take a few steps back in awe of what nature can do. Rubisco allows the Calvin cycle to convert CO2 into sugar. And to me, that is an awesome feat. We could not exist without sugar. My next video will focus on the Calvin cycle that makes glucose, and how it uses Rubisco in its first stage.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.